Hello, dear students. Myself, Dr. Subhash Pawar, Department of Botany, Sant Ramdas College, Bhinsaungi. So, today we discuss about a very important topic of the cell biology and molecular biology, that is a cell division. So, today we discuss cell divisions. Firstly, introductions. Cell division is a very important process in all living organism. During the division of cell, DNA replication and cell growth also takes. All this process, that is cell division. Cell, also called the building block of life, is the most basic functional unit of all living organisms. Cell cycle or cell division is basically a series of events which occur during cell division and replications. In prokaryotic cell, that is cell without nucleus, the cell cycles occurs through a process called binary fission. In eukaryotic cell, that is cell without nucleus. Before a cell can center cell divisions, it needs to take in nutrients. Most dividing cells are double in size between one mitosis and next. DNA is synthesized during only a portion of interphase. The cell cycle is dividing into two basic parts, namely it is interphase and second one is M phase. M phase means mitotic phase. Firstly, we discuss about interphase. Interphase consists of three main phases, that is G1 phase. G1 is cell growth period. And second one is S phase. S phase means DNA synthesis or replication. G2 phase. G2 phase means cell undergoes and prepare for mitosis. Interphase is also known as preparatory phase. In this stage, nucleus and cytosols do not occur. The cell prepares to divide. To divide. That is figures showing cell cycles. Cell cycles is again in four types. That is first one is a first with a growth phase. Growth phase means growth and normal metabolic roles. The G1 phase. Next one is S phase. S phase is a interphase. In this phase, DNA replications and synthesis. This phase is a synthesis phase. Second one, G2 phase, is a growth and preparation for mitosis. Second growing phase. Last one is mitotic phase. In the mitotic phase, again divide four stages. That is first phase stage is prophase, metaphase, anaphase, and telophase. Anaphase and telophase. Okay, first G1 phase. G1 phase means a gap phase. This is the first phase of interphase from the previous M phase until the beginning of DNA synthesis is called G1 phase. It is also called as first growth phase. This phase is marked by synthesis of protein, rRNA, mRNA, and tRNA. During G1 phase is highly variable even among different cells of the same species. Next, S phase. During S phase, replication of DNA and synthesis of histone protein occurs. Histone protein occurs. New histones are required in massive amounts immediately at the beginning of S periods of DNA synthesis to provide new DNA with nucleosome. The amount of DNA in the cell has effectively doubled. Next, G2 phase. This is the second or growth phase of interphase. The cell then enters the G2 phase, which lasts until the cell mitosis. The significant biosynthesis occurs during this phase, and RNA and protein which are required the process of mitosis. M phase. M phase means mitotic phase. This is the last phase. It occurs in the somatic cell and it is meant for the multiplication of cell numbers during embryogenesis and blastogenesis of plant and animal cells. Cell growth stops at this stage and cellular energy is focused on the orderly division into two daughter cells. It is a short period of chromosomes condensation, segregation, and cytoplasmic division. First, mitotic phase. The name mitosis comes from Greek words, that is, mitos means traits. That name was first coined in 1882 by German biologist Walter Fleming and described the thread-like chromosome appeared in animals. Definition. The process where a single cell divides resulting in a formation of two identical cells, each containing some numbers of chromosomes and genetic contents as the form of original cells. Mitosis is divided into four stages. That is, first is prophase, metaphase, anaphase, and telophase. That is, first is prophase. In this phase, due to prophase replication in interphase, each chromosome now passes two chromatids. Both chromatids remain connected with each other by centromere and both remain close associated with on their length. Cutting fibers became coiled into chromosomes and start into coils. The somatic spindle composed of microtubules and proteins in the cytoplasm. The nuclear envelopes and nucleolus start to disappear. The two pair of centroles move away from one another towards the edge of the cells. Next, metaphase. Nuclear membrane disappears. Nuclear membrane disappears completely. In animal cells, the two pair of centroles aligns at opposite pole of the cell. Two centrosomes start pulling up the chromosomes through their attached centromeres toward the two ends of the cells. Therefore, the chromosomes comes under longitudinal tension from two ends of the cells. Chromosomes align at the metaphase plates at right angles to the spindle poles. Anaphase. In these periods, centromeres in each distinct chromosomes begins to move apart. The centromere of each chromosome is divided into two. The centromere is divided into two, and two chromatids. Each chromosome get separated from each other. These chromosomes are separated from to each others. Once the pairs sister chromatids separated from one another, each is considered to a full chromosome, which is considered to the full chromosomes. The daughter chromosomes move to the pole at opposite and appearance of V-shaped. In the daughter chromosomes, is appearance V-shaped here and here, like that, V-shaped. The proteins that bind sister chromatids together are cleaved. Are cleaved. Telophase next. Anaphase and this is telophase. The telophase, the polar microtubules continue to lengthen. Corresponding daughter chromosomes attach at opposite end of the same chromosomes is attached on the opposite end of the cells. Nuclei begin to form opposite poles. Nuclei are form opposite poles. Spindle fibers start to disappear. These spindles are start to disappear. Chromatin fibers are chromosomes uncoils and becomes thread-like. Here the thread-like this reappears. Thus, after rearrangements of chromosomes, two daughter nuclei are formed due to the cytokinesis. After these changes, telophase is complete. Telophase is complete and genetic contents of one cells have been divided equally into two. Divided equally into two. Divided equally into two. Cytokinesis. Next on cytokinesis. In this stage, cytoplasm divides into two and result into formation of two daughter cells. It starts after immediately the telophase is over. In plant, the structure coalesces into a cell plates at the center of the rhagoplast and development into cell wall separating into the two nuclei. In animal cell, cleavage furrow is formed while in the plant cells, cell plate is formed from the Golgi apparatus. The number of chromosomes in each daughter cell is equal to number present in the parent cells. Significance. Mitosis results in the production of diploid daughter cells with identical genetic complement usually. Mitosis causes increase in number of cells within organisms and cell repair. In some part of the body, skin and digestive tract, cells are contextually replaced by new ones. New cells are formed by mitosis. Some organisms produce genetically similar offspring through asexual reproduction, that is, Hydra reproduces asexually by budding. Some divisions happens during asexual reproductions or vegetative propagation in plant. Mitosis or the equational division is equally restricted to the diploid cell only. Thank you. Then next, we will continue the next chapters and next lectures. Thank you.